Good evening, one and all. Today we are going to discuss about structure of chitin. Chitin is basically a modified form of homopolysaccharide, and that is a derivative of uh, we can say it is a derivative of N-acetyl glucosamine. And what is N-acetyl glucosamine? N-acetyl glucosamine is made up of glucose and as well as N-acetyl group, which collectively forms N-acetyl glucosamine, and that is forming a complex structure known as N-acetylglucosamine, and that is basically having a structure of chitin. Chitin is basically the second most abundant carbohydrate on Earth. Second most abundant carbohydrate on Earth after cellulose, and that is having a linkage of beta D glucoses. It is having beta D glucose which are collectively bonded together, but these beta D glucoses are having um, N-acetyl glucosamine on their second carbon. On their second carbon, they are having N-acetyl glucosamine on their structure, due to which this structure is collectively known as N-acetyl glucosamine. And where is it chitin found? Chitin is found in the fungal cell wall. Chitin is found in the fungal cell wall. As it is found in the fungal cell wall, it is also known as fungal cellulose and fungal cellulose, as well as it is also found in the exoskeleton of arthropods, as well as in the exoskeleton of arthropods, shells of mollusks, crustaceans, as well as it is also present in arthropods, crustaceans, as well as in other such structures which are present inside the structure of chitin. These are basically present in the arthropods, in the exoskeleton of arthropods, alongside calcium carbonate, as well as these are also present inside the skulls of, not skulls but shells of mollusks and crustaceans, and these are found in two forms. First is known as the pure form or modified form. First known as pure modified form, and pure form which is unmodified form. Pure form which is unmodified form. This pure or unmodified form is basically uh, not having a thick, not having a tough structure, that is having following characteristic: that it is tough, it's resilient, it's pliable, it's flexible, as well as it's translucent. On the other hand side, this type of structure is present in the fungal cell wall, while when it comes to the structure of chitin, in impure form or modified form, that is quite harder, quite stronger, quite tough, as well as that is not flexible, and that kind of structure is present in the exoskeleton of insects, exoskeleton of insects, as well as in the shells of mollusks and crustaceans, etc. So I guess this is how the structure is formed. As you can see, this is a linkage in between um, chitin, and I know that you people are quite good at identifying the linkage by giving the number of bond. So here we are having this linkage, which is known as beta 1,4 and acetyl D-glucosamine. After which we are having some other MCQ's point that here beta 1,4 and acetyl D-glucosamine represents what? It represents first beta. Beta represents, it is having beta glucose, which is uh, D glucose, dextrorotatory glucose, B glu and beta glucose. And what kind of linkage is there? There is 1,4 linkage, which means the hydroxyl group of first carbon is linked with hydroxyl group of the fourth carbon of the other beta D glucose in order to form a linkage which is known as beta 1,4 D uh, uh, glucose linkage. But here, as we are having an N-acetyl group on the second carbon there is an N-acetyl group, due to which we call it as beta 1,4 N-acetyl D-glucosamine. So ultimately, this is the linkage, and linkage between them can, in simple words, written as beta 1,4 like beta D1,4 glycosidic linkage. After which, this is the N-acetyl group, as well as um, these are the structures, as well as these are basically the structure of chitin is similar, is analogous to the In terms of structure, the structure of chitin is analogous to the structure of cellulose. It is analogous to the structure of cellulose. Why? Because the structure of cellulose is analogous to the structure of chitin is analogous to the structure of cellulose. Because chitin is unbranched, cellulose is unbranched as well. Chitin is made up of crystalline nanofibers, whereas cellulose is made up of crystalline fibers. There's the only difference that these are having N-acetyl glucosamine, while rest there's almost all similarities are same, except the fact that it is having N-acetyl group. And after that, we are having N-acetyl group, and that is functionally the structure of chitin. Functionally, chitin is comparable to keratin protein because it functions as keratin protein, which is present in the hair and nails of humans. So that is comparable to that as well. Alongside that, this chitin is present in fungal cell wall as we previously discussed, and it is known as fungal cellulose as it is written over the screen as well. And what is the general formula for that? General formula of chitin is, it is somewhere something like C3H5O8 C3H5O8N, and then we are having integral number n. That is the structure, but you can google it out further because these are having the general structural formula of chitin. This is a homopolysaccharide, and I guess that's all about this video, and hopefully we'll meet again in another one.